Hello guys, I am Dr. Srijit from Government Medical College Thrissur. Today I will be discussing on the topic electron transport chain and oxidative phosphorylation. We'll be discussing the topic under the following headings: the two donkeys, which we'll see later, the oxidation by ETC, phosphorylation by ATP synthase, and coupling of these two mechanisms by chemiosmotic theory, and also poisons affecting the oxidative phosphorylation, solving how the body solves the problem of extra mitochondrial lineage. So we know that coming to the two donkeys concept, we know in real life donkeys are used to carry loads on their back. So whenever a huge luggage is kept onto the donkey's back, it gets reduced. As you can see from the picture, donkeys gets reduced when the load is kept on their back. Similarly, NAD gets reduced when protons are loaded onto that. Similarly, FAD plus is also reduced when protons are loaded onto that. So these are known as reducing equivalents. NAD plus and FAD plus are known as reducing equivalents because these contains protons. These can be loaded with protons. So when these kinds of loaded donkeys are produced in variety of reactions in our body, both extramitochondrially and intramitochondrially. So what happens in these conditions is that whenever these donkeys are produced, these donkeys with their loads on their back, that is, these reduced equivalents, are carry these protons and electrons to the electron transport chain, which is situated inside the mitochondria in the inner mitochondrial membrane. These complexes will receive the electrons from these donkeys or these reduced equivalents. These electrons are received from them, and the electrons travel down through the whole complexes of the electron transport chain, and finally oxygen is reduced to water. So that is what finally happens in electron transport chain. Oxygen is reduced to water. But when you ask me if why is this occurring, why is oxidative phosphorylation or electron transport chain is operating inside the mitochondria, I will say that it is the main aim of such a function, such a apparatus inside mitochondria is to produce ATP from ADP. Okay, so let's see how body creates ATP from ATP and inorganic phosphate. So that is the main aim of this function, this apparatus in the body. So this is a picture showing the whole electron transport chain as well as the ATP synthase complex, which is known as complex 5, also known as complex 5 ATP synthase, and the other four complexes are also shown. So there are complex 1, complex 2, complex 3, and complex 4, and there are two mobile carriers known as coenzyme Q and cytochrome C. Okay, so what happens is that when one donkey carrying the NADH and H plus, this is one donkey, and the other donkey is FADH2. Both are reduced donkeys. As you can see, protons are loaded onto them. So NADH plus H plus will give their electrons to complex 1, and this complex 1 will give these electrons collected from NADH, and it is given to complex 1, and the complex 1 will give it to complex 3. Similarly, FADH2 will contribute its electrons to complex 2, and 2 will give it to complex 3. And how do these complexes give their electrons to complex 3? It is using the help of coenzyme Q, which is a mobile carrier. Okay, the coenzyme Q will collect electrons from 1 and give it to 3, as well as collect electrons from 2 and give it to 3. Okay, you understood the concept? So this is what is happening in a nutshell, and finally 3 will pass on these electrons to cytochrome C, and then to 4. So 3 will pass to 4 using the mobile carrier known as cytochrome C. So let's see what happens in detail in each of them. So coming to complex 1, you can see that NADH, as I said, NADH will give these electrons to complex 1, which is known as consists of FMN and a series of iron sulfur centers. So that is complex 1. So complex 1, after going electrons will pass through the complex 1, and then it will end on coenzyme Q. So the complex 1 is named as NADH Q oxidoreductase. Okay, so you can name the complex using the path of the electron travel. Okay, so this is what is complex 1 is all about. Now coming to complex 2, as I've said earlier, complex 2 receives its electron from FADH2. FADH2 is produced in a reaction mainly in Krebs cycle, namely when the succinate is converted to fumarate, and FADH2 is produced, and FADH2 will give electrons to the complex 2, which consists of FAD and again a series of iron and sulfur centers. Okay, and again it will give to finally to coenzyme Q, which will collect the electrons and give it to complex 3. Okay, so again we can name this complex 2 from the path of the electron travel, succinate Q or reductase. Okay, succinate Q reductase. It should have been oxidoreductase, but for some matter scientists have chosen to name it as succinate Q reductase. Okay, now coming back to the whole picture, now we have finished 1 and 2. Now we have seen 1 is giving electrons to 3 through Q, and again 2 is giving to 3 through Q. Okay, now we'll see what is happening in 3. Now 3 is supposed to receive electrons from coenzyme Q and pass it through the complex 3, and then it is giving it to cytochrome C, which is another mobile carrier, which will later give the electrons finally to complex 4. Okay, so what is complex 3? Complex 3 is cytochrome C1, Bl, BH, and consists of a special complex known as RISC iron sulfur complex or iron sulfur center. So that is complex 3, and appropriately it has been named as Q cytochrome C oxidoreductase. Okay, so the naming is also easy. You don't have to remember anything unless you know, if you know everything or all the components involved in it. So it is easy to name a complex. So this is complex 3. Now coming to complex 4, we have, you know that complex 4 receives its electron from complex 3 through the mobile carrier cytochrome C. So cytochrome C will give its electron to copper, through the complex 4, which consists of copper A, heme A3, and copper B, which you can see is a combination of heme proteins and copper containing proteins. So complex 4 consists of a heme and iron and copper containing complexes. So the electron will pass through this, and finally oxygen will collect the electron and combine with 2 protons to give water. So the final product of electron transport chain is water. The final reduction process results in water formation. So you can see, as the electron travels down the complex 1 to complex 4, you can see whenever an electron is passed from one component to the other, the previous component will get oxidized. The next component, the electron receiving component will get reduced. So it's a series of oxidation reduction reactions, or you can call it as an oxidation. Complete process is an oxidation. So this part of the whole ATP synthesis is known as oxidation part. Now we can go to the ATP synthesis part. So ATP synthesis part, or the you can call it as phosphorylation part, because ADP is getting phosphorylated by inorganic phosphate and it is converted to ATP. So you can call it as phosphorylation. And coming to ATP synthase, which is also known as the complex 5, you can see in this picture that it consists of here it is the inner mitochondrial membrane, is the outside of the mitochondria, is the inside of mitochondria, and there is as you can see, there is an F4 component and an F1 component. This is the F1 component and this is the F4 component. Okay, this is the F1 component, this is the F4 component, and you can see it has a rotatory mechanism attached to an axle, and it is a gamma subunit as well as an epsilon subunit. Epsilon is not shown here, but yes. So moving forward, so whenever this protons are pumped inside this mechanism, this motor shaped subunit, whenever protons are traveling in this direction, this rotor will start to rotate and it will generate ATP. ATP is generated naturally by combining ADP and inorganic phosphate. So this mechanism, this ATP synthase complex is for generating ATP inside the mitochondria. So this is how ATP is generated inside the mitochondria. Now, scientists earlier in 1950s and all, the scientific community was already aware of the fact that there is some mechanism called electron transport chain happening, and finally water is being formed, and that is the path of electrons being they already knew about the path of electrons, the flow of electrons in the electron transport chain, and there is oxidation occurring, they knew. And they also knew that in another part of the inner mitochondrial membrane there is an ATP synthase complex, and in that synthase complex is able to synthesize ATP using ADP and PI. But they were not able to link these two processes and explain how mitochondria was able to synthesize ATP using this electron transport chain. Okay, so this is where a great scientist contributed to the scientific world, that by making a discovery that there is he came up with a theory known as chemiosmotic theory. This theory explains that uh, this theory explains that yeah, this is Peter Mitchell, and this theory explains that this is mitochondria, this is inner mitochondrial membrane, and all the four complexes are situated here, and there is an ATP synthase complex situated here, as I showed in the previous picture. So oxidation was occurring here, phosphorylation was occurring here. This scientist was able to come up with a theory known as chemiosmotic theory and was able to link these two. And what happened is that what he told is that for every let me okay, so let me redraw this. Okay, so this is complex 1, complex 2, complex 3, complex 4, and this is complex 5. Okay, ATP synthase. So as the electrons travel through the complex 1 to complex 4, what happens is that protons are being pumped into the intermembrane space. Protons are being pumped into intermembrane space. So he also told that for every 2 electrons passing through the electron transport chain, 4 protons are being pumped into the intermembrane space by complex 1. No protons are being pumped by complex 2. Again 4H plus is being pumped by complex 3 and 2H plus by complex 4. So as the electron transport chain is operating, the intermembrane space will be becomes rich in protons as the time passes by, and it will swell up by like anything, and the proton motive force will develop inside the intermembrane space, and there will be a heavy tendency to pass into the matrix of the mitochondria, but the inner mitochondrial membrane is not permeable to protons. So that is when the body utilizes the uh, this ATP synthase complex, and the whole H plus which is accumulated in the intermembrane space gets will be transferred into the matrix through the ATP synthase complex, which will rotate the motor and it will generate ATP. Okay, this is what chemiosmotic theory states. So for that, Peter Mitchell was awarded Nobel Prize in Chemistry in 1978. So that's again, this is the diagram showing the number of protons pumped by each complexes. As you can see here, okay, as you can see, the complex 1 and complex 3 are pumping 4H plus, whereas complex 4 is pumping 2H plus into the intermembrane space. And as you can see, complex 2 is not providing any Complex 2 is not providing any protons into the intermembrane space. So that is a point to be kept in mind. So and that's all about electron transport chain and oxidative phosphorylation. Now we have seen how the body is able to generate ATP utilizing the electrons and protons that have been pumped onto the reducing equivalents, and how electron transport chain and ATP synthase complex is are in a way coupled. You can call them they are acting in a coupled manner. So this coupling was brought about by this chemiosmosis. And now we can explain, we can move forward into the topic poisons of oxidative phosphorylation. So there are several substances which inhibit the electron transport chain. There are substances which inhibit the oxidative phosphorylation, and also there are substances which uncouple these two processes. You know, we have just said that electron transport chain and oxidative phosphorylation is being coupled, and they are called together as oxidative phosphorylation. Some substances can uncouple them. These are known as uncouplers of oxidative phosphorylation. Now, talking about inhibitors, these are the various inhibitors of electron transport chain. And we have inhibitors of oxidative phosphorylation like oligomycin and atractyloside. And there are several uncouplers of oxidative phosphorylation like 2,4-dinitrophenol, thyroxine, thermogenin, etc. Okay. So these are about poisons of oxidative phosphorylation in a nutshell. Now, I've talked about the donkeys in my first slide. The donkeys, I've said that these donkeys carry the protons, the NADH plus H plus, as well as the FADH2. These are reduced forms of their coenzymes, NAD and FAD. So these are generated usually in the mitochondria itself, so they don't have to cross the mitochondrial membrane to reach the electron transport chain in order to unload their protons. But some NADH are produced outside the mitochondria. Okay, like processes like glycolysis and some other processes also. In all these processes, NADH is produced outside the mitochondria, and somehow the body has to find a way to bring the NADH, the loaded donkey, into the mitochondria. So generally there are two shuttle mechanisms operating in our body. One is glycerophosphate shuttle. As you can see, this is a glycerophosphate shuttle, and the other shuttle mechanism is the malate shuttle mechanism. This is the malate shuttle mechanism. Okay. So generally, the NADH2, the malate shuttle is considered more efficient than the glycerophosphate shuttle. So this is all about electron transport chain oxidative phosphorylation. I hope this was a very useful class for you, and all the best for your exams. Thank you.